Today, we talk about nutrition and we have a very special guest. Hello, everybody. Welcome back to the show. Today is a very special day. I want to do this for a long time. Let me bring my guest, a guy that doesn't need any introduction. Randy Oliver himself. How are you, Randy?
Hey, Umberto.
I'm good. I'm good. Let me just check if the comments are coming. So, okay, I think we are live. Let me know in the comment chat, guys, if everything is okay, if you can listen to us, okay. So we're having some technical issues, but we are working on that, right, Randy?
Yes.
Good, good, good. So, Randy, first of all, how are you? It's been a while.
I'm doing fine. Busy. I'm out in California, so we had almond pollination, which means we get a earlier start than most beekeepers. We had a warm winter and intermittent snow. So we had snow a week ago, and now we're 80 degrees today. So the hives are back from pollination, and we're splitting them like mad. My sons are making about 240 nucleus colonies a day, day after day, and to stay ahead of swarming. And we sell quite a few nucleus colonies, and then start all of our colonies fresh with fresh queens every year.
Fresh queens.
Yes.
Good. Randy, let me start. Can we get a? I think everybody here, they know why they're here. I already make a couple videos about that. I received a lot of questions from our audience here, and can I ask you to give us the presentation so we can see your presentation 20 minutes or so, and then I have my own questions, and we start to back and forward about the data and how the study was conducted, etc. Sounds a plan?
Sounds great with me.
All right, so let me share the screen here. Let's see. Let's figure that out if we still have that technical problem.
Okay, I'm changing back and forth between the first two slides. Looks like it's working.
It is working. Yeah.
Great.
So we fixed it. Perfect timing.
Okay, I'm going to start off with this with a trial that I ran in 2020 and then wrote up last year. And as I mentioned, almonds. I'm in California when we have roughly every commercial colony that can be pried off the ground in the United States moved to California to pollinate the almonds. And this is what it looks like out here. It's these huge artificial forests of almond trees that all come into bloom at the same time. Huge value up here, 82% of the world's almonds, over 1 million acres. And so that means over 2 million colonies of bees are in here for a short period. This year, we got released from some of our yards 1 week from when we brought the bees in. So you may get paid for only 1 week. And our stronger colonies, we rented for $230 a hive this year. To put that in perspective, when I started doing it, we got $10 per hive. When I remember hitting $18 a hive and thinking I died and gone to heaven. So just in my career of pollinating almonds, we've gone from $10 per colony to $230 per colony. So it just changed everything. And the growers do pay quite a bit for this service. This is a lot of money involved. And here's some hives placed in a typical young almond orchard right here. These are maybe 6-year-old trees right here. Now here's some of my hives in an almond orchard, and we get paid by how strong our colonies are. So 10% of our colonies get hand graded for cluster size, and then we get paid proportionally to that. So we want to make sure that we meet contracts on all of our colonies. And the thing that I've learned is that to get a strong colony to be graded in mid-February, it has to go into the winter with a large cluster. Otherwise, it doesn't have time to build up in the springtime. It's too late in the springtime to try to make a colony grow fast by mid-February. So you really have to get your cluster. And this is not a particularly large cluster, but this cluster size in the fall is critical. Now, where we live in California, in a very dry summer environment. It's always been a dry summer environment, but lately it's been the worst. Way past my memory, we're right now in the middle of the worst drought that California's experienced in 1,200 years. And it looks like we thought maybe because of some heavy rain in October that we'd be coming out of it, but that's not the case. So we're going into continuing in this drought. This is typical fall, late, or actually from about July on conditions in my area, where we have pine and oak forests. I have more pines in some yards, but there's nothing in bloom for the bees. Only a tiny smattering of late summer, fall bloom. And because of that, for 25 years, I moved my hives out of California every summer up to irrigated pasture in Nevada. And most commercial beekeepers move them out of during the summer. Then I realized I could use pollen substitute and keep my hives at home during summer and manage them better for Varroa by feeding pollen sub rather than depending upon natural pollen. So this revolutionized my beekeeping, being able to feed pollen sub in late summer to stimulate late season brood rearing, to grow that not only the winter cluster, but the winter cluster composed of well-fed winter bees, or diutinous bees, survivor bees, essentially nurse bees that instead of producing jelly, store all their nutrition in their fat bodies and go into that long-lived state to make it through the winter. And those are the bees that then will initiate brood rearing in the spring and build up that cluster for almond pollination. So we feed pollen sub in the fall in any yards that need that, which are most of our yards. And then we're often asked to take our bees down to almonds prior to the bloom starting. So here's an almond orchard here, a young orchard, not a flower petal on the trees. And you notice in many orchards, they have a scorched earth policy. There's herbicides, there's no ground flora at all. So we actually move in late January, early February, from a light nectar and pollen flow up in the foothills down to this desert down here with nothing for the bees to eat. If we took them off of that natural nectar and pollen flow down to this desert, while they sit here for a week or 10 days until the trees come into bloom, they would suppress brood rearing. The nurses would start cannibalizing the brood, and our colonies would not grade well. So here's my sons, Eric and Ian, right here, putting a 2.5 pound chunks of pollen sub at each drop of bees, and they're putting that in there so the colonies will continue to build up until these trees come in bloom. So the big question for us, since we spent a lot of money on pollen subs, we used to mix our own. Then we discovered that some of the off-the-shelf ones are pretty good. But the question always is, and asked by other beekeepers, do some pollen subs promote colony growth better than other subs? So I did a trial in 2013. You can look at my website, where we compared the growth of these colonies from mid-August until February with the control group, not fed any pollen sub, just nosediving, like right here. This is our colony strength, frames of bees, so going down to almost 2 frames of bees, whereas the positive control group, which was fed patties of natural pollen, did well, and then the various pollen subs did almost as good as the natural pollen. So this is very encouraging that we have pollen subs in 2013 that could do well. So I was asked by a number of beekeepers with some new pollen subs in the market, how do the new ones compare? So some beekeepers down in the Bakersfield area were making their own homebrew formulation in very large quantities, truckloads of ingredients. And they were curious how their sub compared to those on the market. So they chipped in some money and said, Randy, would you run this trial? So I said, okay, I will run this trial. So this is their formulation right here. I had them make it up to me and weigh it up into 1 pound patties. I actually checked the weights to make sure they were the same. Theirs is a homebrew formulation. I published the recipe on my website, and it has 20% natural pollen in this. So then I used the standard that we've been using for a number of years, which was the Man Lake bulk soft. We prefer to feed in a bulk form rather than a patty form, and we'll talk about that later. So I knew we've had long experience with this performing very well for us. So then I approached Global and I said, what's your flagship product? And they said, well, we have plain pollen sub, we have 4% pollen and 15% pollen. I said, whichever one you want me to test. So they said they sent me the 15% pollen formulation here. So these two were the only two of all these subs that contain any natural pollen. The rest of these are all completely artificial diets, no pollen in them. Then Dayton had AP23, which I had not yet tested. Mega B, developed by Dr. Gordon Wardell, and when he was at Tucson, on the market still. Man Lake's flagship, Ultra B, although Man Lake, for some reason, changed their yeast in their product in the spring of this trial, or in the summer of this trial, and it was much darker than usual. And I've had some feedback that the bees didn't like it as much, and we noticed that too. They've since changed back, so this particular trial may not apply to their current formulation of Ultra B. And then there's a new one called Healthy Bee, and I've worked with that manufacturer before. And this is a spirulina algae product with corn gluten meal in it also. And but it also had thyme oil added to it for possible varroa control or whatever. That turns out to be kind of repellent to the bees. So this manufacturer now is selling this also with no thyme oil on it. So I have not formally tested that formulation, but this is the one with the thyme oil. And then the question always is, well, how can I tell that it's the benefit of the pollen sub, or is it just the sugar in these patties? So I ran a negative control of just sugar alone, sugar in a fondant, equal to the amount of sugar average in the patties. Then we decided to run this large scale, 144 hives in the trial, 8 types of patties, so 18 hives per patty type. And then we replicated this in 3 different yards to see if we got consistent results from yard to yard. Now, you can understand feeding this many patties out at each feeding could be very confusing. So here's my son, Eric. What we did is we're essentially blinded to treatment. We don't really care about what brand it is. So we color-coded the 8 different types of patties. And before each feeding at home, we would put them into color-coded with tape shoe boxes here, count out the 18 patties, go to the 3 yards. And when we got to the yard, we had all the hive numbers marked with the colored tape for the patty type. And then each of us would lay a tub out on the proper hives. And then a quality control person would walk the entire yard, make sure everything was right. And then we would feed the appropriate patties to each hive. We also fed a sugar syrup too. This is a typical yard. We chose our 3 worst yards that historically we knew that if we didn't feed pollen sub, the colonies would nosedive. Here's my 2 sons, Eric and Ian again. Typical, very dry conditions. We did a randomized block design. So we took the 8 strongest hives in each yard. We graded them all for strength, took the 8 strongest and fed them, randomly assigned one of the 8 different pollen subs. Then the next 8 strongest randomly assigned the subs. That way I could see if there was any effect upon colony starting strength or not. And we turned out there wasn't. And we fed them all throughout the summer, took a brief break, and then started again in the winter. Here's the timeline of the project right here. The orange squares are feedings of pollen sub. The blue circles are feedings of sugar syrup. We didn't need, anytime we saw that there was not fresh nectar in the cells, we would feed near the brood nest, we'd feed sugar syrup. And actually, by this point in time, late in the season, they were getting plugged out with excess syrup. So there was no lack of sugar anywhere in the trial. So we prepared the colonies here, first grading here, midpoint grading here, and final grading here, feeding pollen sub all during this period, except for 2 yards, which had a very surprise yellow star thistle flow that we had not expected for about 10 days. So we stopped feeding the sub in these 2 yards only until the flow was over. And the colonies brooded up suddenly. And then as soon as the flow stopped, they stopped brooding up. Then we had this long dearth period here. In November, the colonies shut down brood rearing. We didn't feed them. And then it warmed up, surprisingly. Normally, it doesn't warm up in December, but it did. And so colonies were interested in taking feed. We started feeding again, and we could feed up to the point up to 21 days after the first natural pollens came in, which came from alder. After 21 days, any bees that emerged may have been affected by the nurses eating this alder pollen. So we counted 21 days from the first pollen, and we did our final assessment right then. We fed ad libitum, which meant that we fed when the most attractive subs were just getting nearing the end of consumption in the hives. We fed all the hives all at the same time. So nobody ever ran out of pollen sub at any time other than the winter break. And then we would remove and weigh any uneaten sub to keep track of that. The bottom line is that all the subs were pretty well eaten. At least 90% of all the subs were eaten. So nobody was ever hungry for their pollen sub. To our surprise, despite the fact that in the previous year we were able to increase colony size by feeding over the summer, we were unable to do that during this trial. The colonies actually mostly shrunk in size. And it may be due to this big difference between the 2020 weather and the 2019 weather, which is on the dotted line. So the 2019, when the colonies grew, we had nice, very cool weather. These are daily highs here, dropping down in the 60s and 70s during the fall. The colonies normally will brood up very nicely with that. We had temperatures up to 100 in October during the trial. So this very hot weather was stressful to the colonies. And also the humidity. So here's our humidity low, which was very, very low during the trial and much higher during buildup period the previous year. So these may have had strong effects. So what I have here is a graph of the change in colony strength with one time means that from August through November, there was no change in strength. So that would be this line. Any dots above this line, they grew. So very few colonies grew, notably these red ones, the homebrew formulation, and all the rest of the colonies shrunk. And of interest is compared to our starting strength, the stronger starting strength colonies tended to shrink more than the smaller starting strength colonies. So just contrary to what you might have expected. And I'll tell you right now, one thing we've learned with field research over the years is throw everything that you figure out will make sense or that's practical sense, throw that out the window, because things like this will always surprise you. The colonies that started larger were the ones that lost the most strength as opposed to the smaller ones. But you can start to see here by looking at the colors. Look at the whites, the control group. They're all well down here. And the greens, the spirulina ones, are all well down here, whereas the other patty types are more up here, staying the same or growing. This is a typical frame in mid-October, showing they had plenty of syrup around here. They all had a box of honey on top of them. Nice brood patterns, but not one speck of pollen in the combs. So the only protein they were getting were from the pollen subs. And here we are at our midpoint grading in November with my newest technician here, my grandson Ryder, who was out of school due to COVID, and very bright kid, and turned out to be an incredible field technician, making sure as quality control, checking every hive number ahead of us and making sure we did everything right. And here we are grading the colonies for strength, looking top and bottom for each box and counting the seams filled full of bees, and Ryder recording that data. Okay, so here at the midpoint, we look at colony strength. So we started off at about 8.5 frames average across the board. As I said, most on average, they shrunk. The two outstanding ones were the global and their homebrew patties here, compared to the sugar controls. Now notice the error bars, how small they are. I was way surprised. Normally in a field trial, you have great big error bars, but when you have this small of error bars, that indicates this is significant differences here. So the question then, as far as how they performed, let's look at the protein content of the subs. My collaborator, Dr. Vince Rosigliano, I sent him samples of the subs and of bees, in which he then had analyzed for nutrients, and we'll get into his analysis of the bees themselves. You can see the subs range for wet weight anywhere from up to 22% protein down to about 14% protein, but no correlation with performance with the subs, because this was one of the better performers historically and in this trial right here, and these two are the best performers. The fat content, this is always of interest. People say, Randy, how much lipid, how much vegetable oil should we have in pollen subs? So for the wet weight, although the recommendations are often up into that 3 to 5% range up here. The best performers were two of the lowest lipid contents, with the global clear down here to just a 2%. This is 2%, 0.2% lipid content. So questionable how important the lipids or the sterols are in the pollen subs. Sugar contents, we had them analyzed for sucrose, glucose, fructose, and then total sugars is the colored column here. And you can see the two top performers were in the middle there. So it does not appear to be that the sugar syrup was important. And again, the sugar content, we fed sugar throughout the trial. Now here's very interesting. Vince took my samples of bees I sent to him, and then dissected away the heads and thoraxes individually, and then dried them, and then weighed them. And here's the total mass of the heads and thoraces. The thoraces indicating muscle mass, and the head indicating the glandular mass. So these two protein tissues right here. So here's your on the left, the column on the left is your head and thorax mass. The column on the right is the total frames of bees for the number of the hives in each group. Notice how closely they correlate. Okay, that it's very the weight of the bees very much reflects the overall performance in colony strength, indicating that this was a nutritional issue. Clearly, that affected the total population of the bees. So individual bee nutrition also was reflected in total number of frames of bees for the test group. This then to next-gen sequencing on the gut microbiome. So these are the core bacteria found in honeybee guts worldwide, every colony. And I was curious whether the feeding artificial diets would change the structure of the community structure of bacteria in the bee gut, and also in the total abundance of bacteria. So this is total bacterial abundance in the bee's guts. And you can see in the global patties here with the 15% pollen had the highest abundance, which you would expect. But the homebrew with 20% pollen had one of the lowest abundances. So bacterial abundance, and these are totally artificial diets. So most of the artificial diets fell between the two that contain large amounts of pollen. So bacterial abundance did not correlate at all with the amount of pollen in the patties. So yeah, here's 15% pollen, here's 20% pollen right here. The other thing to look at is the community structure. Notice the amounts of the different bacterial types. That also was consistent in all. So these bacteria, which have evolved over millions of years to live in the bee guts and bees eating natural pollen, they adapt very well, apparently, to the artificial diets. Another big surprise. Then we also had some bees that had colonies that had not been treated with oxalic acid at all. Most of we treated with extended release oxalic acid. So continuous exposure to oxalic acid for most of the colonies during the trial. And what we found of interest in each yard, we had a few untreated colonies, which were potential breeders for varroa resistance. And I compared the gut microbiomes of them, and it looks like oxalic acid also does not have any substantial effect on the gut microbiome. That's good news for us. Okay, so then in mid-December, we initiated pollen subfeeding again. Here's one of these spirulina patties right here. And you can see there's no natural pollen in the combs, but the bees did initiate brood rearing well. And final grading was on February 1st. Very, very cold morning out there. So the cluster sizes probably underrepresent what they would be graded at in the almonds. So here's typical for one of our yards of the performance over the course of the year with each type of pollen sub, with this being the 20% natural pollen, 15% pollen here, this one with no pollen, and none of the rest with any pollen. And the sugar control showed in the black right here, where those colonies went way downhill from that 8.5 frame starting strength down to 4 frames, not even big enough to take to almond pollination without combining them. And these ones here are actually growing slightly larger than they had started with last August. Colony loss. Well, if you look at over here, this is your percent of colonies lost, censoring out any that went queenless. And the sugar controls, they just dwindled away to nothing. So we had over 30% of those colonies did not make it through to almonds, whereas all of the ones that were fed the global, and most all with the homebrew, made it to almonds. And then here we have the same thing again. This time I have the no change diagonally right here, so it matches here. And what you can see is about half the colonies ended up from their November strength to their February strength growing during that period of time, during the December and January feeding. And about half of them shrunk further. But again, you really see the difference in the colors. Look at all the red and blue colonies up here, and look at all the white and green colonies down here. So a big difference in performance. And just to show you a histogram where we have colony strength from 0 frames of bees up to 12 frames of bees. We did have some strong ones. And the number of colonies in each group of 18. We have just a normal curve of the control group right here, and almost no overlap with the homebrew group. Average strength less than 3 frames here at the end. Average strength up here of over 8 frames of bees here. So big difference due to the feeding of pollen sub. And when we did the final assessment, we get paid about $25 per frame. So we combined the weak colonies. So what I said, well, let's just look at how many frames of bees we had to go to almonds. So out of each group of 18 colonies, this is the total number of frames that actually we were able to take to almonds from that group. And there are 3.5 times as many bees in the homebrew group as there were in the sugar control group. We found that to be very interesting. So now the question is, why did some subs perform better than other subs? And we get to the concept of Liebig's barrel. Now imagine that each of these staves in the barrel reflects an essential nutrient in your diet. One might be vitamin C, one might be vitamin A, one might be iron, one might be zinc, one might be tryptophan, essential amino acid, one might be phenylalanine, essential amino acid. In a perfect diet, all the staves would be right at this top level, and you could fill the barrel, you get optimum growth. But if you have one essential nutrient at a low level, that's all the higher you can fill the barrel. That's all the more you can grow. So despite an abundance of other nutrients doesn't help at all, because its total growth is limited by the amino or the essential nutrient in the lowest level right here. And that brings us to the state of our art. This is, if you ask anybody about honeybee essential amino acids, they will refer you to this study, this paper. It's about an 80-page paper by Antonius de Groot from the Netherlands, published in 1953. Okay, I've been married to this paper for many, many years because it's a fantastic paper, incredible researcher, huge amount of work. But he was very clear. His work was a pioneering work that needed interpretation and open to interpretation, which I will now get into, reinterpreting his findings. This 1953 was the beginning of animal nutrition, where researchers for the various animal industries were developing the amino acid ratios for animal nutrition. Nowadays, if you're raising pigs or you're raising chickens, you can buy a formulated diet for each stage of growth. And for each stage, they change the amino acid ratios for optimal growth. We got nothing like that for honeybees. Everything we know about essential acids for honeybees essentially is from this one chart, is figure 15 right here, where he made he fed emerged bees in an incubator, emerged adult bees in an incubator, a diet of sugar and a mixture of amino acids to mimic casein or milk protein, which is a complete protein. And what he did is he would take any one amino acids. For example, here for valine, he only fed, if he limited that to 0 in the diet, the colonies, this is the amount of growth here. The bees did not gain weight. If you put in 1%, they did gain weight. If you put in 2%, they gained even more. 3% gained even more. 4%, they gained, but 5, 6, 7%, no additional gain. So this was the inflection point here. So I said, relative to the other ones, it's a waste of money to have more than 4% protein in that diet. It just so happened that tryptophan came in at 1%. So he normalized everything else to tryptophan and made a ratio relative to tryptophan, which turned out to be, I think, a big mistake. Because when we use de Groot's recommendation, a percent of essential amino acids in a diet, I put those in as the black columns right here. Okay, notice that all the other essential amino acids seem to be in abundance, surplus above the de Groot's ratios here. But there's two ways to look at this. You could either say they all have excess of these amino acids, or you might say, wow, they maybe have a deficit of some relative to these really high ones. And this is what I'm looking at. I'm curious about this large amount of excess leucine right here. The reason is when we look at de Groot's breakdown, that he says that 16% of the essential amino acids should be leucine, going way down to only a small percent for tryptophan. To me, it makes more sense to say, wow, we should normalize it to leucine, not to tryptophan. The other thing is that he also found out that when he fed the diets that were deficient in one particular amino acid, both the increase in dry weight and the increase in nitrogen content, which indicates how much was actually incorporated into the body of that amino acid, leucine stands out in both of them as one of the most critical of the amino acids right there in a diet. The colonies without enough leucine just nosedive. So now I've reworked the analyses right here, normalized to leucine. So leucine set at 1 right here. Look at some of these deficiencies relative to leucine. This was an incredible eye-opener to me, that it's looking like we have like a huge deficiency of valine in many of these amino acids. And look back here. Let's see what the most critical amino acids essential are. Valine, threonine, leucine, and histidine. Okay, so let's take a look here. Histidine, threonine, and valine. Again, huge deficit in valine, deficit across the board in threonine, except for the 2 best performers. Across the board, deficient in histidine, except for the 2 best performers. And even over here, the 2 best performers had less deficiency in valine than the other poorer performers. Again, what an eye-opener this was for me. So what I did is I just added up all these relative deficiencies here to this level right here. And, oh, I should come back to that. I'll come back to that in 1 second. I made up an amino acid calculator here. So you can put in any formulation, the green column, and compare it to de Groot's recommendation right here. Now, I can put in analyses for royal jelly, which would be a perfect bee food, and it's very close to de Groot's recommendation. Although you need a little more lysine in royal jelly, but other than that, pretty close. A natural mix of pollens, known bee nutritious pollens, very, very close to the de Groot's recommendations right here. So here what I did is I analyzed all the diets from the most productive to the poorest performing down here. And I marked here, to make it easy for you, with appropriate sized red dots where there were deficiencies. The best performing diet had very few deficiencies. The second best, the global, had a couple of deficiencies, but not too bad. And the more deficiencies, the poorer the diet performed. So I added them up and I charted them out like this. So this is your ending total frames of bees per treatment up to that 140 figure again. And here's your total of the deficiencies relative to leucine in the diet. and look at how closely they track to colony performance. And my guess is without the thyme oil, healthy bee would be right up about here. We'd probably have an R squared of maybe 0.9, which means that this means 85% of this variation can be explained by this calculation here. That is, in a field trial with honeybees, that is unheard of to have that high an R squared value. So this means it's probably not a fluke. This really is something most likely. So now what I have is I have a free amino acid calculator that you can download, free download off my website. This is already being used by beekeepers around the world. I've sent the results to all the manufacturers. I not only have the growth suggested ratio, but I have come up with my own suggested ratio targeted towards nurse bee nutrition rather than emerged bee nutrition. And you can go ahead and try to balance a diet out right here. And I put in royal jelly for comparison in the green. So notice now I've adjusted my lysine up here to match what is needed for royal jelly right here. And I just did a little play one. This is what the calculator looks like. And I did a simple, I just punched in 50 pounds of soy flour and 20 pounds of brewer's yeast. And the ratios were a little bit off. So this lets you calculate out. You had to add 3/4 of a pound each of lysine concentrate, threonine, and valine concentrates off the shelf you can get, and then come up with a perfectly balanced diet. Using two very inexpensive components and a little bit of essential amino acid balancing. And okay, that's it for my slideshow. So Umberto, we can.
All right, I'll take from here. Randy, thank you very much for the presentation. I think in the name of everybody here, to thank you for your hard work, your passion for bees. You know, we need that kind of passion in a beekeeping community, bringing these trials and information to everybody. And I don't need to remind everybody, for free. So go to the website, scientificbeekeeping.com, and check out those resources there. Randy, let's have a chat.
Yeah.
I follow you for many years. We discussed this before, so I would like to dig in and pick up your brain in a couple details that I got my attention.
Okay.
From the first trial, comparing with the second one, can you tell me a little, if you have information about the differences? If there is any difference from those diets that was the same brand in the two trials, because in a trial number one, you have even other brands, some of the brands that didn't perform in the second one performed very well over there, and so there is a chance something was different in the diet. And I know how hard it is to keep things consistent when you're producing in large amounts and you need to get nutrients from various sources around the world. So it's super hard. I've been there, not easy. But do you have information about these two trials, the differences between those patties?
That is a very good point. And with the Ultra Bee, which had performed very well before, they did change the yeast. And I'm not the only beekeeper who noticed a difference. Other ones have also corresponded with me. And again, the manufacturer has changed back. So it's tough. If you're running a trial with something you can't control, you're buying it off the shelf, you are stuck with that.
Yeah, that's a problem. I don't think we have a solution for that. Is, to keep something consistent, or you do yourself and make sure you get your resources from a unique place that never change, which is almost impossible.
Mm-hmm. And here's the other thing: is if you're going to change your formula, you got to run some field trials to validate that you can do that. The point I'm making here, Umberto, is that we have all these different pollen subs developed, and they're different in every country depending upon what raw materials you have available. And I call it the by guess and by golly formulation, where somebody thinks, oh, well, this should be good, and I'll balance that with some of this, but they don't use any frame of reference, or it's totally different than any other livestock industry, which actually has everything analyzed and then balances it out on paper and adjusts things. Where what we see is a lot of these subs are developed by again, by guessing, by golly, and oh wow, this seemed to work pretty well. So yeah, we'll do this. I'm hoping to shift our industry to especially with the essential amino acids you see, which seem to be a huge predictor of performance, that we may see some improved subs coming up soon.
Oh, I'm looking forward for that.
Yeah, and what
Randy.
interesting thing was that last one I showed you of just the soy yeast one. That's one of the least expensive diets you can make right there.
Yeah.
And if you can simply add just a couple of dollars worth of amino acid concentrates to a large batch and bring it up to top performance, we might be seeing some, which is what I'm interested in, because I spend thousands of dollars to buy pollen sub every year, that we might be able to get cheaper subs that perform better.
Since you bring that up, so a lot of these conclusions is being made because of the second trial, right? And the second trial where you have the opportunity to measure the amino acid content and go a little deeper.
Yes.
How comfortable are you with those conclusions in a trial that you didn't see high performance?
I'm not quite sure on your sentence, right? Or your question.
You start with 8 frames of bees and end up with less in the second one.
Yeah.
In the first trial, we start with 5, and most of them at the end of the trial end up with more than what they start with.
Right.
So the second trial has something going on there.
Right.
Can we agree with that?
Oh yeah.
So,
we had severe weather conditions.
Yes, and then the whole system, the whole conclusions and calculations are being made in a trial that was not optimal. So that's my question to you: how, if you see any, if you should be careful with any of this,
Umberto, if conditions were optimal, I wouldn't have had to feed pollen sub at all.
No,
if conditions were optimal, you wouldn't have to feed any pollen sub at all.
Yeah. From the practical beekeeping perspective, yes. But
that's what I do. I do practical beekeeping perspective.
I know. But you also this time did ratios and things like that. So that's why.
I can understand it. But go back to your question.
Yes.
There's no reason to feed pollen sub if things are optimal. We want to feed pollen sub that does the best it can under the worst conditions. And we got the worst conditions there for that. The other point, Umberto, is I didn't analyze absolute values. I didn't compare absolute values. I compared relative values.
Fair enough. So that's those
are very good points you're bringing up. I'm glad you give me a chance to explain them.
Yeah, that's what I was. That's the at least that's my answer for that. And so it is fascinating, right? As you mentioned before, in other industries, we know exactly what we need to give it to them at every single time point of their development, and that fascinates me. And I think we're just starting in that direction. And to be honest, I don't know if this is a good thing or not. So there is always that discussion about how far we know with this knowledge. But I'm a scientist. I want to know everything.
Yeah.
So,
and for me, from a practical perspective, yes, us too. California is becoming a much more difficult place to keep bees. What's interesting, Umberto, is I had a friend here in my own town, been keeping bees as long as I have, and he just retired. And he said, "Randy, I got a big old stack of pollen traps, to get rid of." I said, "Yeah, I'm curious about that. We don't use ours anymore." He goes, "Yeah, me neither. Back in the day, Randy, we used to fill the drawer of the pollen traps in a day and sell bulk pollen." And I did too. Both of us did. We both stopped doing it because we don't get anywhere near that much pollen anymore during our long extended drought. Now we just don't have that much pollen production. So what we're seeing, for me to stay in business, for my sons to stay in business as beekeepers, we can no longer depend upon the natural environment. We either have to put our bees on trucks with a huge carbon footprint and haul them over the Sierra Nevada up to irrigated pasture somewhere, or haul in pollen sub here and keep them at home. So this is just a reality, this business decision for us. How can we best feed, what artificial diet or what diet period can we feed our bees that allow us to continue business to pollinate the almonds?
Yeah, beekeeping definitely is getting harder and harder. At the same time, that is bad for I like to see a bright light in every situation. When you have these deserts of pollen, and and you have a guy like you over there that can do this kind of trials, these are kind of the perfect situation for that kind of trial.
Absolutely perfect situation.
So that's the only bright side that
This,
I can
this is
see.
one of my criticisms of a number of published papers, trials on artificial diets.
Yeah.
That they either don't keep track of the natural pollen coming in, or they're doing it while there's a natural pollen flow coming in. And then they draw conclusions despite having a natural pollen flow coming in. And I found very clearly in my trials, if there's a natural pollen flow coming in, the bees just suck the sugar out of the pollen sub and toss it out the door. They're not interested in that. They just take the sugar out of it.
Yeah.
So you need a special circumstance. Yes. Right now, I'm working with Vince. Now he is Dr. Elton Herbert Jr. Worked in the Tucson lab in the 60s and 70s, and on bee nutrition. And he had developed a system of having flight cages for nucleus colonies. So you put a nucleus colony in a flight cage. You have a whole bank of these, and then you feed that nucleus colony artificial diet, and you see how many rounds of bees they can rear on an artificial diet. He never got more than 2 generations of bees before they ran out of some nutrient on that. That's the perfect way to test. But he died unexpectedly at a young age, around 1980, and nobody picked up that work. I've been trying to get any USDA research group to pick up on that work for years, and I finally found, I hope the guy, and Dr. Resigliano, who was my collaborator in this one. He right now has his first flight cage based upon Elton Herbert's research, set up at the bee lab, the USDA. Just put it up last week, and then that tornado came and blew it over. So he just put it back up again. So if he can get that, duplicate that, and build a bank of these, we can start making serious progress. I'm extremely excited to be working with USDA, what I think is one of the most important research projects now for commercial beekeeping, to develop artificial diets. That he's no longer dependent upon finding a desert. He can do it in flight cages with short-term trials. See if you can get a diet that you can rear 3 or 4 generations of bees on an artificial diet.
Yeah, that's a good thing to pursue, that for sure, Randy. Randy, how about we take some questions from the people? I can see a lot of people writing a lot. I can't follow, so I'm gonna ask you guys to ask right now. If you have questions for Randy, that's the time. And Randy, I'm gonna put here the questions, and we go through one by one. Okay?
Great.
All right. The Canadian Beekeepers Blog.
Bacteria would increase with additional added pollen that hadn't been irradiated. Additional pollen, I'm not seeing that as something. And I got to tell you, I just got analyzed the data today from by another researcher who was asking for my help, who compared colony growth before almonds, feeding either the global 4% or the 15% pollen sub, natural pollen in there, and there wasn't any difference between them. So adding additional pollen, I'm not seeing would help it.
All right, and we continue with the same.
Okay.
My friend Jan in Canada.
No, there was another reason why he did that. He early on in his research, tryptophan kept popping up, and then when it came up with that one, he just went to it. It's very easy for him to do it. Yeah, and I just, again, I have nothing but admiration for all of his research. He put in plenty of qualifiers, and maybe if he would have thought about it longer, he would have normalized to leucine instead. So yeah, you don't always get it right the first try.
That's true. That's why in this channel, I always advise people to see if there is repetition. It's only one paper, just look, that's fine. But, keep in mind that in science, we need repetition, we need confirmation, we need different groups trying to do the same thing and get the same results. And that's the way you move forward in science. Randy, people love you. Just a little example. So here we go.
Okay, great. All right.
Okay, so let's see. If I see, I have more questions here. More questions. Let
Yeah,
me
in answer
see.
to this one, while you're looking, it's
Okay.
just it was amazing. I had no idea we would learn so much from this trial. I thought it was going to be a simple thing. We just take down the numbers of colony strength. It was going to the next step and having the analysis done and then doing the deep dive. That turned out to be the fascinating part of this whole trial. Okay, would it pollen sub perform better than feeding pollen that was collected? That's an excellent question, and that's actually I didn't cover that. That's my research for this coming year, and the reason is that where that pollen is placed. And now we get down to the behavior of the nurse bees and what the environmental cues are to the nurse bees that trigger behaviors. The main behavior being that people think that the queen determines how much a colony grows, and that's totally erroneous. It's the nurse bees that call all the shots because they control the food supply. The queen can lay 1,000 eggs a day here in my environment in August, and the nurse bees follow behind her and consume 1,000 eggs a day. Don't even try to feed them. They just they cannibalize those eggs and recycle the protein in them. So it's the cues from the environment that the nurse bees receive that tell them whether or not they should feed those larvae from the eggs or cannibalize the eggs, whether it's the right time for the colony to go into growth mode. And that is a lot to do with exactly where that pollen is placed. So here's another misconception. Anybody reads the beginning bee books and they say, what's the typical brood frame look like? And you go, oh, well, there's a band across the top of honey and going down the sides. Then you have an oval of brood here. And between that band of honey and the oval of brood, you have a crescent shape like a rainbow of bee bread. And people assume that that rainbow is where the forager bees place their incoming pollen. And that's a totally erroneous conclusion because that's only survivor bias right there. That's only the bee bread or the pollen that was not consumed. That is the same as most of us have a pantry somewhere in the house, and back on the back shelves are the cans of canned food that have been sitting there for way past expiration date that if we were desperate, we would eat. That's what that band of bee bread represents. That's the leftovers that are there just in case they need them. But that band is not stimulatory to the nurses to rear brood at all. And you can tell that very easily by colonies going into the winter with a whole bunch of bee bread stored. They don't rear brood. That's not stimulatory. Stimulatory pollen is pollen that's placed right in the middle of the brood area, which is where the forager bees preferentially place it. Right as soon as a bee emerges from the brood, they pack that cell full of fresh pollen, and it's preferentially consumed by the nurse bees before it's ever made into bee bread. You just don't see it because most beekeepers don't look carefully enough.
Interesting, Randy. Randy, I have two questions that since you brought the pollen back, the two subs that come on top have natural pollen, and when we have 15 in one and 20 on another, are the pollen consistent? You have pollen all the time, or this? What exactly that means to have 20% of natural pollen and 15% of natural pollen? Are they the same nutritional value each pollen? Can we assume that? If so,
no, you can't assume any of that. Yeah, so that I know one of the guys who collects that pollen and sells that pollen. It's a mixture of pollens, but no, that's not an ingredient like buying soy flour or brewer's yeast, which are very consistent from batch to batch. Bee collected corbicular pollen is not as consistent.
Yeah, I'm always curious when because sometimes we have pollen, people collect pollen and give it back, and nothing happened too. Maybe it's a bad quality of low nutritional value pollen. That happens sometimes.
Yeah, like dandelion pollen.
Yep.
Colonies will die if fed only dandelion pollen because it's short on a couple of essential amino acids.
Yeah. Question for you.
Okay, you showed correlation and some error bars, Did you run ANOVA? No, I didn't even bother to run ANOVA. We didn't have that. We only had 18 colonies in each group. If yes, what were the significant differences between ending growth and decline values? Tell you what, Vince took the same data and published a scientific paper, peer-reviewed paper on this, and he focused on other things, on the statistics. So you can download that one. It's a free download, and read that for those values.
Let me put it here in the chat so people know where to get it. If I can paste this somehow. Uh-oh. 1 second. Okay, I'm getting there, I guess. Here we go. Now you guys know where to go and to get the paper.
Right.
It's an open access paper, so everybody have access. Don't need to pay for it, which is always good. Okay, let's see other questions here for you, Randy. All right. What do you know about?
Here's what I see here. Are there enzymes in natural pollen not found in supplements? I would imagine so. I imagine there's enzymes in there.
Yeah, I would assume that as well.
Yeah. But the point is that in all my work with this, I don't have any indication that natural pollen is necessary in a bee diet at all for good performance. We can get very, very good performance on some of these artificial diets. So I'm very curious to see. I didn't run a positive control in the 2020 trial of patties of natural pollen, which again would depend upon, the right mixture of pollens to compare to that homebrew. But that homebrew formulation performed very well. My guess is if I had run a natural control, it would have been right there in the same ballpark as the natural pollen.
All right. Well, I think this is for me. To summarize some of your comments, are we being fooled that bees are taking pollen sub and instead just in taking the sugar out of it and ejecting the rest? Well,
you got to listen to every word in my sentences. That happens if there's a natural pollen flow on at the same time. The bees will then take the sugar out and kick it out. And again, there's no sense feeding pollen sub if there's a natural pollen flow on. It's a waste of money to do that. When there is no natural pollen flow, it can make a huge difference in colony performance, and they eat every bit of it. And I've used fluorescent tracer to check this out, where I mix pollen sub with a fluorescent tracer that glows under black light, and then put screen bottom boards on the hive, and then feed it to the bees. And then you can go out at night and look under the hive in the darkness and out in the front of the entrance and see if there's any fluorescent tracer there, which shows very easily whether they were kicking any of it out. And that doesn't happen if there's no pollen flow on, but it definitely does happen if there is a pollen flow on.
All right, Randy.
Reading comments on how excited everyone is to add pollen or use pollen sub. My interpretation is that balancing the amino acids is the key. That's certainly, as far as that would certainly be the first step, I would say, in an artificial diet. Because it didn't seem to be the lipids, it didn't seem to be the total protein content. I did an experiment the year before indicating it's not, the sterols, it's not critical to get the 24-methylene cholesterol in there. And you got to realize if you're putting in soy flour and brewer's yeast, two basic components, the brewer's yeast is already packed with vitamins and minerals and all kinds of good things. And there's also the soy flour has a lot of things besides just protein in it. Those are both raw foods. Seeds of soybeans have a lot of nutrition in them. So you're getting a lot of those traces already in there. We could fine tune eventually by putting in vitamins and minerals and stuff, but I'm not sure how important those are.
Randy, question, Mike, from you. Do you think this is a constant, the reason, or that varies with whatever the bees need at a specific time? Because maybe a different ratio is needed for, because in a specific time of the year or some conditions, there is a necessity for different amount of different proteins more expressed than others, and then the ratios will change.
Let me ask you this question, Umberto.
Yes.
Who processes all the pollen? Who consumes all the pollen in a hive?
The nurse bees.
The nurse bees, only the nurse bees. So just rule all the rest of the bees out. What's the one thing that the nurse bees do with that?
They feed the others.
They feed it convert it to jelly.
Yep.
So that's the only question. Jelly is consistent throughout the year.
I agree with that.
Okay. Does that answer your question?
So you believe is a
I don't tell you my beliefs. I asked you, does that answer your question?
That giving me more to think about.
Oh, that's a weasel out.
Okay, so
yeah, jelly is consistent throughout the year and from continent to continent. It appears that nurse bees have dialed in. They don't adjust the jelly depending upon nutrition. If they can't put out a complete jelly, they just stop. They put out less jelly or no jelly at all, but they won't put out a substandard jelly. So that should answer your question.
The jelly is constant. Yeah, we have a lot of evidence of that. Many different publications on that.
Right. All kinds of pollens are not created equal. I do not know what pollen was added to those two different pollen subs. Which actually were pollen supplements.
Yeah. So let's see if we can figure this out together.
Bluff flower is the best overall pollen if you're going to plant harvest for pollen. Almond is definitely one that's very good. Most of the rosaceae, the apples, peaches, those kind of plants. Dr. Geraldine Wright has looked at that, which plants appear to be good. But generally, if you talk to any experienced beekeeper in your area, you can tell. And you can't trust the books either. One thing I learned in that 2013 trial is when the alders started blooming in January, after I had colonies that were absolutely starved for protein and down to almost nothing. So I knew there was not a trace of protein. And when the alder pollen came in, I checked it under the microscope over and over. There was no other species of pollen coming in. So they had a 100% diet consisting only of alder pollen. That's a wind-pollinated tree pollen. And what do all the books say about wind-pollinated tree pollen, about the nutritional value? They all say it's not as nutritious. I can tell you, I got photographs. The bees, it is absolutely nutritious to them. The alder pollen, the bees brood up spectacularly on the alder pollen. So you got to be careful about what people assume in the books. Just because it's a wind-pollinated plant doesn't mean that it can't be very nutritious to the bees. Same with corn pollen. Corn pollen, everybody says, oh, it's wind pollen, it's no good. Actually, in the studies where they fed corn pollen to the nurse bees, it actually does do pretty well. Okay, how consistent can a pollen patty be if the pollen is variable? Well, you know the answer to that. Don't waste our time.
Yep, it's not. So, okay. All right, I think we got here, Randy. I want to see, do we have a message to the beekeeping community, the beekeeping world? This is going to be registered forever in the internet.
I don't know if there's any message. I mean, I'm not out to sell anything. We're, we're commercial beekeepers. I want my sons to be able to succeed. California is in a long-term drought. We humans are screwing up the world with climate change. We're experiencing it tremendously here in California, and we need honeybees to pollinate our crops. It's a business opportunity of a business that we enjoy doing. So I'm interested in for my own and my son's self-interest to figure out best methods for beekeeping, and I'm happy to share it with everybody. And that's why I don't. All my research is funded only by beekeeper donations. I don't have any obligation to any company, and I'm not interested in making money on any of this. Just I want to just serve the beekeeping community.
And we all appreciate your passion
Thank
and
you.
your attitude, Randy. Randy, one more question. In the next 10 years, what do you think is going to change most in the beekeeping industry?
Wow, I'm hoping mite resistance in my selective breeding program
Yeah.
for Varroa resistance. I diddled around for, since Varroa arrived, thinking prior to Varroa, I found it was very easy to breed for bees that were any color, bees that were gentle, um, bees that could fight American foul brood, bees that could resist tracheomite entirely, bees that can resist some of the viruses, bees that could resist Nosema ceranae. Those things are all easy to breed bees for. And then when Varroa arrived, I thought, oh, this is going to be easy. I'd already figured out how to breed for all those other traits. And no, Varroa, I wasted about 10 years, half-assed breeding for Varroa, thinking it's going to be easy. So then 6 years ago, we decided to really get serious about selective breeding for Varroa. So we start about 1,500 hives on our own each year, grafted solely from completely mite-resistant colonies that show Varroa does not build up in them. And we're finding out it's not an easily heritable trait. We're going to have to shift the entire breeding population of our bees. We pretty much control our breeding population, our drone pool, in order to get the critical alleles evenly distributed throughout the entire breeding population, because it appears that you need to have there's recessive traits, which means that not only does the queen need to carry the critical allele, but enough drones that she mates with also have to carry critical alleles to match. So a double recessive expression of that gene then into some of the patrolines. So it may only take 2 or 3 patrolines of daughter queens in a colony. So if the queen mates with 40 drones, there's 40 different patrolines of sister workers from each father in that colony acting as a team. It may only take 1 or 2 of those patrolines to exhibit certain behaviors to interrupt Varroa reproduction. So what we found is we started out with 0.2% of our colonies exhibiting resistance. We do very strong selective pressure. Out of those 1,500, we breed off of about 25 a year. So only 25 out of 1,500 colonies pass their genes on to the next generation. Now, of course, the drones come only from the mothers. So that means the genetics the next year are provided from the selected mothers from the previous year. So you're not only breeding the maternal population, you're also, if in enough years, you're also supplying the drones. And that allows you some diversity. Otherwise, if you only selected 25 out of 1,500, you'd bottleneck and lose diversity. But because each of those queens have mated with a number of drones, you have a diversity going on. So we haven't seen any inbreeding effect yet. Anyway, we went from 2%, 0.2%, up to around 2%, up to around 4%. And this last year, we were running close to 15% of our population exhibiting complete resistance to Varroa. We had one yard from one queen mother that 24 out of 48 colonies exhibited Varroa resistance. That's a magic number. That's a magic number because that's 50% of the queen's offspring, which means we may have finally gotten some of those genes into a maternal line for the first time. That's called progeny selection. If it's a double recessive gene, you've got to, and the queen carries that trait, then you can have up to 50% of the daughter colonies exhibit that trait. So those colonies are all marked, and they're going to go to an isolated breeding yard this summer where there's no other drones, and we're going to inbreed that population right there.
How many generations are you right now?
This will be our 6th generation of serious selection.
Okay. All right. That's promising. Looking forward to know more about that.
Oh, Randy. Yeah. So we can't claim mite-resistant population yet. We had 140 mite-resistant colonies that went through the winter out of the roughly 1,000 that we took to almonds. But when we get up to 95%, then we can start talking about actually having grow-resistant strain of bees.
Interesting. So one more time, Randy, I want to thank you one more time. I think in the name everybody in the chat room right now, we have almost 200 people watching you right now live. Thank you for all your effort. Yeah,
my pleasure. There's so much still to learn in beekeeping. It's very exciting.
Yeah, and I'm looking forward to see you again soon.
Thank you, Humberto.
All right, guys, thank you for coming, and see you guys in the next video inside the Hive.tv, the show about bees. See you guys later. Bye.